First, we are going to find all GWAS variants of type 2 diabetes. We'll start with creating the node of type 2 diabetes. Then, the quick add function gives us hint on the connected nodes. We are going to choose GWAS association from a sequence variant to type 2 diabetes. We could submit this query and get a list of results. We can also use export to obtain the complete query results. Then, let's give more restrictions to the GWAS variant. We want to get all variants that locate in the enhancers. We need to add a node for the enhancers and add an edge between them with the label locate in. We could also submit the query now, but we would like to include a bit more criteria. Many enhancers regulate the distal genes by physical interactions in 3D space. Therefore, we want to find the examples when enhancers form 3D chromatin loops with certain genes. In order to do this, we need to add two more nodes, a gene and a loop, and then add two overlap edges from the loop to the gene and the enhancer. We can submit this query and get the example's results now. In this example, the SNP on the X chromosome is located in an enhancer and also forms a loop with the gene ATP2B3. We could even make this query more complicated. For example, we can require the sequence variant to be the eQTL of the gene in the pancreas. By submitting this query, we can obtain an example result. This SNP on chromosome 16 is the eQTL of gene IRX3. It is located in an enhancer and forms a chromatin loop with this gene. This can serve as an example of 3D interactions that play an important role in gene regulation.